Hello, everyone. Thank you for joining this installment of Boyce Hobson Institute's Breaking Ground discussion series. There's a lot of people out there joining us today. It's really nice to see. My name is AJ Bushy, and I'm in the communications department here at BTI. And for this month's Breaking Ground, we welcome Gary Blissard, a virologist here at BTI, whose lab is focused on understanding the biology and pathology of viral infections and interactions in insect cells. And today, we'll be talking with him about his work studying insects and their viruses and how they can help fight plant pests and prevent human diseases. Hey, Gary.
Hi, AJ. Great to be here.
Yeah, thank you for doing this with us today. I really appreciate it. And before we get going, I have a few housekeeping announcements, kind of the basics if you joined us before. During this session, which will be about a half hour, everyone will be muted out there, and Gary and I will have our discussion, and then we'll have a Q&A session, and you can put your questions in the chat box, which I'm sure everyone is familiar with at this point in Zoom. And I've also enabled the live transcript function, so you should be seeing closed captions, and if you'd like to turn that off, you can do that under the little live transcript button down at the bottom of your Zoom as well. And thanks again to everybody out there for joining us today. And I'm really excited for today because Gary is a little unique for Boyce Hobson Institute. Most of our researchers are plant scientists, and Gary is a virologist. And he looks at insect viruses that infect insects. So could you tell us a little bit about yourself, Gary, and how you came to study this interesting topic?
Sure. Yeah, so I grew up in Birmingham, Alabama. I was always interested in biology and went to Auburn University, and there I got kind of interested in both microbiology and entomology, and then I sort of came to insect viruses from a forest entomology course that I took. It was a really interesting course, and I was first kind of exposed to the sort of concept of insects that have their own diseases and that they affect sort of the populations of insects in the ecosystem. And I just got very interested in viruses. And so following my degree in entomology from Auburn, I joined the Peace Corps. And in the Peace Corps, I was a citrus entomologist trying to develop biological control in the citrus industry in Belize. And so I was working on various topics there. And when I finished my time in the Peace Corps, I went on to graduate school to do a master's degree in insect pathology. And in fact, I did my degree with a young assistant professor named Brian Federici, who was in fact a postdoc at Boyce Thompson many years earlier. So I kind of knew about
a familiar
Boyce
name, yeah,
from way back. So when I did my master's degree there in insect pathology, and then went to another lab to do a PhD degree, a lab at Texas A&M, the lab of Max Summers, who was one of the large labs in the baculovirus research. And I had kind of focused on baculoviruses as a group of viruses I was interested in because they're very pathogenic and important viruses in biological control. But in fact, when I went there, I worked on a different kind of virus, a virus called polydnaviruses, which are genetic elements that are embedded in the genomes of parasitic wasps. A very fascinating topic, but I won't talk about that today because I haven't followed up on those studies, but really interesting topic. So there at the very end of my PhD, I started working a little with baculoviruses with the expression vector system, which a system that had just been developed at that time. And then I moved on to do a postdoc at Oregon State University with a guy named George Rohrman, who is well known in the baculovirus field. And I started working on structural protein genes there. And from my postdoc there, then I came to BTI and started my lab working on gene expression in baculoviruses. And then I developed another project to work on main project, which has sort of carried through my whole career to work on envelope proteins of these genes.
Could you tell us a little bit more about baculoviruses and why you're interested in them?
Sure. So baculoviruses are these very broadly distributed viruses in nature, and they infect mostly lepidopterans, which are moths and butterflies, and they cause a very severe disease. Now, the moths and butterflies they infect, many of them are very important pest insects in agriculture, so they have a huge potential for use as biocontrol agents. They're also very specific. They generally infect a single species or very few.
So Gary, I just want to jump in real quick. We're not seeing your slides if you're sharing.
Okay, yeah, I had not shared it yet. Sorry about that.
That's no problem.
That now?
I thought we were probably close to getting into the slides, so I just wanted to.
And
yeah, there you are in Belize.
so have you got my slides there?
Yep, we see them.
Yeah, this is just a picture of sort of my time at Belize and the Peace Corps. Very fond memories there. Our research station on the left here. This is a young me in my early 20s and the staff of our research group there at the Citrus Research Station and some citrus there. Most of the citrus there is grown by small growers that just have a few acres and they have their house there. So the Mexican fruit fly was a big disease problem, sorry, not disease, but a pest there. And we were working on parasitic wasps that were capable of injecting their eggs in and killing the larvae of those insects.
Right. It's like a biocontrol.
So we were talking about baculoviruses, getting back to the topic here.
Yeah.
So I've just shown here some pictures of baculoviruses that are important ones and their hosts, the beet armyworm, fall armyworm, African armyworm, and some of the baculoviruses that infect them. And in many of these pictures I've shown sort of the healthy larva and the sort of end of the disease state, and the disease from a baculovirus often results in just the liquefaction of the larva, and that liquefied mess there is essentially a pool of virus that is then transmitted to sort of the next insect that comes along, or the next susceptible host. One we study is a virus called ACMNPV. It has a very long name, Autographa californica multiple nucleopolyhedrovirus. That's why we call it ACMNPV. And we use as a model an insect, the cabbage looper, which is Trichoplusione, or I'll refer to it as teeny in subsequent slides. Mm-hmm. So the infection by this sort of is instigated by a form of this virus called occlusion bodies. And these are sort of like spores. They're large crystals that within them contain, embedded within them, are the infectious virus particles. And so when an insect, a susceptible larva, usually a lepidopteran, eats these, this large occlusion body dissolves in the gut and releases those infectious virus particles, and they then make their way to the cells that line the gut, the epithelial cells of the midgut. They infect those cells, and then the infection moves into the rest of the animal. It becomes systemic and infects almost all other tissues of the host. And so for this virus, we can kind of subdivide the infection into a primary phase, the gut, and a secondary phase, all the other tissues. And that's kind of illustrated in this slide. So we study this virus and its infection in the gut, and then we also study in other tissues. And so when the gut gets infected by these what we call occlusion-derived virus particles, which come out of these occlusion bodies,
And those are like the kind of the harder, like spore-like crystally kind of form.
yes, and they're very resistant to sort of the insults of nature, sunlight and desiccation, those kind of things. They can persist in the environment for long periods of time. So
Mm,
we consider them environmentally stable. And so when the
gotcha.
animal eats them and it releases this form of the virus, it enters the cell, the virus replicates, makes new progeny, and these little rods um, are very characteristic of this form of this virus. Virus, in fact, named baculovirus, is named for baculum, the Latin baculum, which means rod, is named after these structures that form. And so these are basically called nucleocapsids. They're little shells that contain the genome of the virus. Well, they migrate or they're transported to the basal sides of these cells where the virus then buds out into the open circulatory system of the insect. So this form of the virus is different from the virus that infects the cell. It's called the budded form of the virus. It has different proteins on its surface, and it kind of performs a different function.
And
So
when you say it buds out, it's what exactly do you mean there? It's taking part of the cell membrane with it?
yeah, so it pushes through the plasma membrane of the cell, and it picks up these envelope proteins as it's doing that. And so the envelope of this budded form of the virus comes from the plasma membrane of the cell. And I'll show you a little bit later how that's different from this occluded form of the virus. So that we think of these two forms of the virus, the occluded form of the virus, it has its role in nature is to spread the infection from animal to animal. And once the animal becomes infected, it makes this budded form of the virus, which it spreads the infection from cell to cell within the infected animal. So two very different functions. And at the end of the infection in the secondary phase, these cells make huge quantities of the occluded form of the virus. And the animal breaks down and it releases them into the environment. And that sort of completes the circle of the infection cycle. So you were talking about pushing through the membrane. This is an electron micrograph actually taken by Bob Granados, a scientist who was here at BTI. This is 40 years old. And this was before we knew anything much about the genome of the virus or what genes did what. But they were studying this by electron microscopy, and you can see that soon after infection, these nucleocapsids are pushing through the basal surface of these cells. So in the secondary phase of the infection I was talking about, where other cells are infected, those infections are initiated by the budded form of the virus. And I know this is complicated, but I just want to make a couple of points here. One is that it's initiated, this phase is initiated by the budded form of the virus. The virus moves into the cell, it uncoats or releases its genome into the nucleus, and then the virus replicates and makes new nucleocapsids that are filled with a genome. And some of those leave the nucleus and make more budded virus, which helps move the infection from cell to cell. But some of those nucleocapsids also stay in the nucleus and become enveloped there to form this occlusion-derived virus particle. And then a protein called polyhedrin accumulates around those and then eventually crystallizes to make those occlusion bodies. And there's a massive amount of this occlusion body protein polyhedrin that's made. And this is actually the basis of the expression vector system that's used in biotechnology very widely. So this virus is very easy to engineer. And so one can replace the polyhedrin gene with a gene that makes the protein that you would like, for example, a vaccine protein or a therapeutic or that sort of thing. And then you can use insect cells in this baculovirus system to make large quantities of this protein. And it's a very effective and productive way to make proteins.
Right. And that is done today, right, with some BTI derived cells.
Yeah, so BTI has been involved in making cell lines that are useful for this kind of process. So I was talking about how much of that protein, the polyhedron protein, is made. These are just some pictures of cells that are infected with a baculovirus, showing the
Oh, wow.
polyhedra, or the occlusion bodies that accumulate, and they just fill the cell. And that protein can be as much as 40 or 60% of the total cell protein by the end of infection. And this middle panel just shows if you purify these, the virus we work with, the one called ACMNPV, makes cuboidal occlusion bodies. And this on the right shows some sections and electron micrographs through occlusion bodies, and you can see the nucleocapsids and the ODV, or occluded virions, within them. So those are the infectious units of the virus.
And so this is the form that it accumulates and then kills the cell and makes the caterpillars all melted and gross, like you showed earlier.
Yeah, so the whole process of the cell dying and lysing and all is very complex. That kind of leads to another topic, is this is a pretty complex virus. These are considered large viruses. So in the world of virology, some viruses are fairly small, maybe encoding 3 to 5 or 10, 20 genes, whereas baculoviruses are considered large viruses. The virus we work with, ACMNPV, is about 134,000 base pairs long. It's a circular genome and encodes about 156 genes, depending on how you interpret data that exists. So the infection cycle is also kind of complex. It starts with an early phase where some genes are expressed. They turn on late genes. Late genes then lead to another set of genes called very late genes. And that all leads to the production of these two virion phenotypes, we call them, the occlusion-derived viruses that are in the occlusion bodies, and the budded form of the virus, shown here. And they both have the same genome, but this genome has a specialization for making these two forms of the virus, and also for taking the virus through this journey in its life cycle, through the primary phase of infection and through the secondary phase of infection. So our studies of gene expression have sort of, or have focused on sorting out some of these questions. For example, what differs about the early and late phase, but also the primary and secondary phases of the infection cycle. And so for many years, we studied baculoviruses sort of one gene at a time, like everyone in our field. But with genomics and transcriptomics, we've been able to now look at the whole genome and transcription and gene expression across the whole genome at once. It's very powerful technology. So I'll just show you an example of some recent study, fairly recent, in which we looked at the expression of the baculovirus in the primary phase and compared that to its infection and gene expression in the secondary phase. And so for the secondary phase, we use cell culture because that mimics, or it's a proxy for the secondary phase, because those cells are infected by the budded form of the virus and produce a lot of the occlusion bodies. For studies in the primary phase, we do this by infecting animals and dissecting out the guts and doing transcriptome studies in the gut. So just a sort of a quick picture of sort of the kind of data and kind of analysis we do something called ranked expression profiling. So looking at each gene in the genome and what its expression profile looks like at a specific time after infection in either the midgut, the primary phase of infection, or in the cell line, which is the secondary phase of infection. So, for example, on this graph, each label here represents one of the 156 genes of the genome. And so the expression profile of that gene in the cell line is shown by an orange dot, from the highest expressed gene to the lowest expressed gene. And then we overlay on that the profile of each gene's expression in the midgut. And so we're looking for how the expression of these genes differs in these different phases of the infection cycle in the animal. And these circles just circle those that fall outside. The gray area is just sort of the noise level we consider. And those that are far outside this are those that are dramatically different in their regulation in the midgut compared to the secondary phase of infection. And so this just shows 12 highlighted here, 7 that are dramatically upregulated or increased in their expression in the midgut, and 5 that are dramatically downregulated in the midgut. And then we can look at those genes and ask, first, do we know what they do? And second, if we do, does it make sense that what they're doing in the infection cycle based on what we know about their function? And I'll just show you a couple of these, or 3 of these, as examples of genes that we did know already from prior studies what these genes did. So for example, a gene called FP25K was dramatically downregulated in the midgut. And it was already known that if you knock out this gene and look at it in cell culture, then the knockout results in an increase in production of the budded form of the virus and a decrease in the occluded form of the virus, or the occlusion bodies. And as you can see, in the midgut, that makes a lot of sense, and that's
Right.
What the virus needs to do to move the infection from the gut cells into a systemic phase, or the secondary phase. Another gene called VFGF, that stands for viral fibroblast growth factor, and this is a homologue of an insect hormone that recruits. It can cause recruitment of other genes of the insect to the sites of the infected cell, and that results in releases of metalloproteases and caspases. These are proteases which then degrade the non-cellular basal lamina layer that surrounds the gut, and that allows those budded virions to escape. So sort of a nice picture is now evolving of how the virus accomplishes this. Another gene called ARIF1, this is actin rearrangement inducing factor 1, causes actin to rearrange within infected cells. And we know from prior studies in other labs that actin is kind of the motor that propels these nucleocapsids in cells. And so that actin, this ARIF1 gene, we think its upregulation of these cells probably helps to push those budded viruses out of the infected cells.
So this is pretty like a sophisticated virus, it sounds like. It's almost like viruses in one, and it depending on where it is and when, it's turning on all these different genes to really do like multiple different things.
Yeah, the virus, with 150 genes, the virus does a lot. I mean, not only is it encoding all the information to reproduce itself, making the structural proteins, but it also manipulates the physiology of the host and even the behavior of the host. So it's there's much to be discovered here about. So we know a lot of phenomena that we don't quite understand how these things work, and now using these sort of modern tools, we can actually start to get at a lot of this.
So you mentioned some of these other. Here you brought up the envelope proteins. How does the virus know when it buds? How does it know to bud on the right side of the gut cell and not just go back down into the lumen there?
Yeah, great question, because that's actually the topic of our other main project. So when I first started my work here at BTI, I was working on kind of the structure and function of these envelope proteins in the budded virus. It has one major envelope protein that's a receptor binding protein, and it's a fusion protein. It's involved in getting into cells. But in the last I don't know, 5 to 10 years, we've been looking at issues of transport of this protein within the cell. And it's really important that that protein shows up on the basal side of the cell. So during the infection, this protein called GP64, that's shown by these kind of red dots and the spikes on the budded virions, it's transported in a polarized manner. Now these cells are polarized. They have an apical side and a basal side, and the protein, once it's produced, moves to the basal side, not just anywhere. And that's where the virus buds through. So this kind of question of how envelope proteins from viruses move in a polarized manner through an insect gut cell, that's a question that's of really broad interest, because a lot of different insect viruses need to do that. So, for example, viruses like arboviruses that are vectored to humans and other mammals by mosquitoes, they have to accomplish the same goal of moving through the midgut cell, and their envelope proteins have to do the same thing so that the virus can bud through into the circulatory system.
And the arboviruses, that includes diseases that like human diseases, right? Like dengue fever.
Right, many human diseases. So we work on a model called vesicular stomatitis virus and its envelope protein called the G protein. And we're also developing work on the dengue virus envelope protein and the Zika virus envelope protein, as well as chikungunya, another arbovirus that's of importance in human pathology.
Right.
So plant viruses also, there's quite a number of them that are transmitted by insects that also do the same thing. They're taken into the gut and have to move through the gut cells. In most of those cases, it's a little bit different because they don't have the same, most of the ones that are studied don't have the same kind of envelope proteins and the same, don't have exactly the same issues, but it's still the same problem. And we're trying to develop an experimental system that will allow us to address that in all kinds of different viruses, as well as pathogenic viruses like baculoviruses. So I'll tell you a little bit about some of the work that we've been doing there. So this just sort of recapitulates that problem. This is reversed, where the apical side, the gut side, the gut lumen side is on the top here. But the virus has to interact, enter the cell, replicate. It makes its gene products, or the components of new virus particles. They have to assemble and bud out the other side. And so our particular focus is on these envelope proteins that are made and transported to the basal side of the cell and then coordinate with the rest of the virus to bud out. So the way we've addressed that is more recently, we've developed a system using sort of two different approaches. One is an Rnai screen in cultured cells, and another is to make transgenic insects that express these viral envelope proteins. And for these studies, we've chosen a Drosophila system because Drosophila is a very powerful genetic and molecular model, and there are many tools that we can draw on to answer the questions that we want. So I'll tell you a little bit about each of these just very briefly. And first I'll talk about these Rnai screens in cultured cells. So the approach we've taken was to look for the host genes that are required for transport of a model envelope protein, GP64, to the surface of cells. So there are a number of different pathways that cells use to move their membrane proteins to the cell surface. And viruses don't recreate everything. They utilize systems that already exist in the cell. So in this system, we first generated an engineered cell line that can inducibly express our model envelope protein. So we integrated this envelope protein gene under the control of inducible promoter. And this allows us to just add copper to the medium of the cells to turn on expression of this envelope protein. And so the way we did this RNAi screen, or this experiment to find the important host genes, was we plate these cells in a multi-well plate, as shown here. And in each well, we add a different double-stranded RNA to target a specific host gene. And so that allows us to knock down the expression of that gene. And so after we incubate that for a little while, we then add copper to the media to turn on expression of the envelope protein. So then we measure the level of the envelope protein at the cell surface. So if the double-stranded RNA in a given well interferes with that trafficking of the protein to the cell surface, then the levels at the cell surface are reduced in comparison to our control. So we can quantify that by flow cytometry and then do some quality control experiments to make sure we're not looking at artifacts or side effects.
Yeah. So you're basically, in each well, you turn off a single gene and then add copper, and that induces the envelope protein to be made, and then you can see if it gets to the end goal or not. Or
yes, does it get to the surface?
Right.
And so, we looked at, in this screen, we looked at 213 genes that were known to be involved in trafficking or movement of envelope or membrane proteins. And the results from that, I'll just show you. I'm not going to go through all this, so
okay.
don't worry. We initially found about 37 that were 37 genes that when we knocked them down, they substantially reduced the amount of envelope protein at the cell surface. But when we did all our controls, we narrowed that down to 6 important genes. And 3 of those are Clathrin or Clathrin adapter proteins. And Clathrin is a protein that's involved in sort of the formation of small vesicles that are involved in moving proteins around in cells, particularly in the secretory pathway and in something called endocytosis. So 2 others were Rab proteins that are regulators of that sort, that process called vesicular trafficking of proteins, and they're small GTPases that are known regulators. And there are in Drosophila there are 29 of these Rab genes, and they each have very specific functions. So by identifying the ones that cause this effect, we can kind of narrow down where the pathway of movement of this protein is in the cell. So this is just kind of the summary, and we haven't, by a screen like this, we certainly haven't answered all the questions, but we've developed some really very good clues as to what's going on. So basically, after what we can suggest from these studies is that after the production of this envelope protein in the ER and it transports through the Golgi, the protein is then moved either through secretory vesicles or through something called fast recycling endosomes to the cell surface, and it accumulates at the cell surface. And so in the same study, we also found that some of the envelope protein, some or maybe a lot of it, is endocytosed back into the cell, and most of it is probably trafficked to recycling endosomes and then back to the cell surface. And this sort of recycling of proteins is very common for membrane proteins, and many other viral proteins are known to do this. Another interesting thing that we saw in this study was that a very small quantity of the envelope protein is released from the cells in something called exosomes. And we don't know exactly what the function of that is or the total significance of this is, but it's something to look into in the future.
Hey, Gary, I'm just keeping an eye on the time. We want to make sure we have enough for a Q&A, and I know you want to talk about the transgenic flies, and then maybe after that we'll hit the Q&A if folks want to start putting the questions into the chat box.
Okay, so I'll go through this kind of quickly. The last bit I was going to talk about are the transgenic flies. And so basically for this, we rely on the expertise of our collaborator, Nicholas Bouchon, who's a Drosophila geneticist. But we've been able to build fly lines that allow us to express our model envelope proteins. And I just show VSVG here in the gut. And so we use something called a GAL4 UAS system, which is a common system that's used in Drosophila. But this allows us to express only in the midgut our envelope protein. And in fact, we can turn on that protein when we want. So using a repressor of GAL4 called GAL80, a temperature sensitive mutant. So basically we can grow these flies, and when we want to do our experiments, shift the temperature and turn on the expression of these envelope proteins. And so this just shows the results of some of our earlier experiments where we express the VSVG protein or the GP64 protein. And just to orient you, this is the gut lumen and the hemolymph. So the virus is normally trafficking in this direction. But when we express these proteins by themselves, no infection, the envelope proteins traffic to the basal surfaces of these cells. So that means the proteins have all the information they need to interact with the machinery of the cell to get to the right place. So that opens up a whole, a large world to us. We can now screen for host genes that are important, and we can also look at signals in the viral proteins that direct this trafficking.
So it's pretty much the insect proteins are shuttling the bacterial envelope protein to the right side of the cell.
Right. The baculovirus is using the trafficking pathways,
right?
And cells have trafficking pathways already existing to move proteins to one side of the cell or another, because there are many host proteins that are doing that. And this just shows one experiment which shows that we can actually knock out one of the host cell proteins using RNAi in a fly line to knock down that protein, express one of our envelope proteins, and we can see that it disrupts that trafficking. So this basically shows that we can knock down host genes that are important in trafficking and identify the host genes that are important. And so
that's the red. The red is what you're looking at?
So this is a different color scheme in the last slide. The envelope protein GP64 is the red, and it's at the basal side of the cells in our control. But when we knock down that host gene, you see it's no longer localized to the basal surfaces of those cells, but it's distributed throughout. Right. So we're looking at a lot of different host genes, and as well as signals in viral envelope proteins. And as I mentioned earlier, we've incorporated other envelope proteins such as those from dengue and Zika and chikungunya into our current studies. And we're also developing tools to do similar things in mosquito guts. So move the technology and the results that we can produce in Drosophila into the relevant host species or vector species. Right. So with that, I'll just end there and tell you about the people who actually did the work. The work on global gene expression from the baculovirus was done by Anita Shrestha and Yunru Chen, with great help from Silin Zong. And our collaborators on that project are San-Yun Fei here at BTI and Ping Wang at Cornell, who's San-Yun is a genius at bioinformatics, and Ping Wang is an expert at the Trichoplusione gut. So they were both really important collaborators on the project and on ongoing projects. So the work that was done in the Drosophila system was done almost single-handedly by Jeff Hodgson, a very talented postdoc that works with me and Nicholas. And I didn't talk about any of his work, but a new student on this project, Robin Chen, I'd just like to give him a shout out because he's doing really good work now, and we hope to get some really great results out of this project. And of course, our very close collaborator, Nicholas Bouchon, who's really the Drosophila geneticist and a expert on insect midgut. So with that, I'll finish, and if there are questions, I'd be happy to talk about.
All right. So thank you so much, Gary. And if folks have questions, you can put them in the chat box. There aren't any in there yet, but I have a couple more. And one is about, so baculoviruses, you mentioned they infect lepidopterans, right? So that's mainly butterflies and moths. And there are a lot of those pests in agriculture, right? They eat up a lot of our crops. And are baculoviruses used to control them now? Is that a thing?
Yes, there are a number of baculoviruses that are used. One is the virus of Helicoverpa armigera, and so that's a pest in Africa, Australia, South America. It's very widely distributed and a devastating pest. It consumes many different host plants, and so the virus for that has been developed as a biocontrol agent and is being used really extensively in Australia and Brazil. Brazil has also used they have another pest called velvet bean caterpillar, which is a pest of soybean, and they've used baculovirus against that for many years. They use baculoviruses in organic apple orchards against something called a codling moth. There's a very effective baculovirus. A lot of studies done on that virus.
Okay. And you mentioned that these are really specific for the host, for the insect, right?
Right. Typically, a baculovirus will infect, only one or a few known hosts. Not one exception, but an exception is the virus we work on. It tends to have a fairly broad host range, and it was sort of selected by the fact that it grows well in culture, and that may have something to do also with its host range, its broader host range. We're actually very interested in that, and one of our ongoing projects is to look at this virus in what's called a semi-permissive host, an insect that can get infected, but the infection isn't very successful. And so we have a project to look at a comparison of the infection on the transcriptome level in the permissive host, the one that we work with, the cabbage looper, and compare that to Helicoverpa zea, which is a semi-permissive host, but an important insect.
And we have one question from David Silverman. Have you seen insects used in your experiments that have developed any immunity to the viruses to which they've been exposed?
That's a good question. No, we haven't. Over the years, there have been a number of studies to try to look at that question, and it's fairly difficult to develop immunity or resistance against baculoviruses. That's always a concern. Like there's in insect populations, there are resistance to BT and certainly resistance to insecticides. It's something one has to watch out for and address. And so when in the laboratory setting, when resistant strains of insects have been selected just by infecting a colony of insects, infecting them, taking the survivors, growing those, infecting, growing, one can generate resistant insects, but they lose that resistance very quickly. And it's thought that the reason for that is that insects that are resistant likely develop a very thick, what's called peritrophic membrane, which is the lining of the gut, and that probably prevents the virus from getting to the midgut cells. And but the problem is, the reason that resistance is lost, and this is all just sort of hand waving in theory, but it's thought that that's lost because it's a very, there's a very heavy price to pay for the insects that have that thick membrane. It probably causes them malnutrition and they can't absorb nutrients as efficiently as other insects. So once you remove the selection pressure of the viral infection, they lose the resistance.
That makes
So
sense.
that's kind of what's known by me.
And that sounds different from, you mentioned BT, that's Bacillus thuringiensis, right? Which
Right.
bacteria that creates a toxin. It seems like that's probably a different type of resistance, being resistant to a toxin is a different mechanism, I would
Yeah,
imagine.
so BT, yes, is a widely used biological, but it is based on a toxin. And so that's very different from in a baculovirus. There's not a single protein that's produced that kills the host. It's the infection, it's the 156 genes coordinating a very sophisticated attack on the host. So, as we all know from studies of COVID and SARS-CoV-2, understanding how a virus interacts and causes pathology is very complicated and difficult to understand.
Right. And speaking of COVID, some of these T&E cells, the insect cells that you work on, they're used to make some vaccines and things for other diseases. Is that correct?
Yes, I briefly mentioned the expression vector system, but that's really widely used in biotechnology. There's literally thousands of labs that use baculovirus to express proteins, and it's widely used in the pharmaceutical industry as well. So, a couple of examples right off the top of my head is the HPV vaccine against cervical cancer, the one that is sold. There's by the one that uses a baculovirus to make the vaccine is sold by GlaxoSmithKline. It's called Cervarix, and so that's been around for a number of years. But there's also a company that's called Novavax that's made a vaccine against SARS-CoV-2, and that's made in a baculovirus system, and that's one that's it's right on the brink of being approved by the FDA for use in the U.S.
Mhm.
And there's a lot of proteins and therapeutics and vaccines in the pipeline made in baculovirus system.
Yeah, it sounds like a lot of the proteins that use some human cells, they have a lot of people don't like using them for ethical reasons. It sounds like as well. So I imagine using insect cells, you don't really need to worry about that type of issue.
Yeah, one of the big advantages of insect cells is they're not susceptible to infection and contamination by human viruses. So this has been a problem in production of vaccines and other things that are made in human cells. If the cell line that you're using has an endogenous virus or gets infected and produces something that might be harmful to humans, that's a bad thing. And and usually they take it off the market and have to go clean it up or whatever. But these insect cell lines are not susceptible to most viruses that infect humans. There are a few crossovers, but not much.
Makes
So
sense.
it's kind of a safety issue that makes them highly advantageous. And that combined with the fact that they make enormous quantities of protein makes them very attractive for production. The other side of it is that they process proteins. They're post-translational modifications of proteins proteins get glycosylated and phosphorylated and modified. Well, all those modifications are very close to what occurs in human cells. There are some differences, and in some cases they're important, but in many cases the proteins that are made in insect cells are very good for vaccines. Sometimes that's even an advantage.
Mm-hmm. Well, that's fascinating. All right, are there any other questions out there for Dr. Blissard? I'll give folks another minute or so if you want to put something in the chat box. Oh, I think that's it. Oh, we got one more from Paul Debbie here. The first ever vaccine for human use made in an insect cell system was made by GSK in a cell line developed by BTI. Yeah, that's right. So that was the one you were just talking about, right?
That was in a cell line called Hpv. That was made by a scientist here, or developed by Bob Granados. And yeah, that was a very interesting story. And we've actually worked with a number of derivatives of that cell line and all the transcriptome work that I was talking about, that was done in one of those derivatives of the Hpv cell line. And they're very productive for protein expression, in comparison with other cell lines.
I guess I should throw it out there. If there's anyone out there who wants to make a lot of protein in an insect cell, let us know. Our tech transfer folks have some ready for you and some paperwork. So you could get those. We have another question from Robert Manel. Does the fact that the buds use the animal cells help it transfer to other cells in the host? Question.
Yeah, I think I know what you're getting at. Are the membrane proteins from the cell that get into the virus particle somehow having a function in the next stage and infecting the next cell. And that's something that really there's nothing known about. In general, the concentration of the viral proteins is so high that it crowds out most of the host membrane proteins, or many of them. But it is known that if you're co-expressing another envelope or membrane protein in high quantities in the same cell, sometimes those can be picked up by the virus particle. Now, as to whether it's a viral infection, it is possible, but I don't know of anyone who's ever looked at that experimentally. Good question, though. It's very interesting. Maybe that's a new research project.
Is it known how the virus organizes its internal communication system so that sequences occur in a given manner?
Actually, that is also an excellent question. I've always been fascinated by the infection cycle and communication within the infected animal. So somehow, all the tissues, as the infection begins, all the tissues in the animal don't get infected systemically at the same time. But somehow the virus is coordinating its infection so that dissolving of the animal and the maximum production of those occlusion bodies occurs and is coordinated. Now, there's very little known about it, but it's not haphazard. It's, because you would think that the first cells that get infected in the hemocoel might go to the end of the infection cycle and lyse and just cause a mess, whereas other cells are not infected. But it's to the virus's advantage to somehow coordinate it so all of that happens at the end, with most cells producing more virus. And that happens, but no one really understands how it happens. So maybe something like this, the hormone, this FGF I mentioned, or other homologs of host cell hormones may have similar effects. It is known that the development of the host insect is in fact manipulated by the virus. there's a gene called ecdysteroid glucosyl transferase, and ecdysone is the molting hormone of insects. And so an insect can escape the infection by molting into the pupal stage. So the larva normally goes into a pupal stage, and ecdysone is the enzyme that regulates that. And so the virus makes an enzyme that modifies the host hormone and prevents it from going into the pupal stage.
Wow.
And that allows the insect to grow a little larger, and it can then make more virus particles. So in using baculoviruses in biocontrol, one of the things that was done in Lois Miller's lab many years ago was to knock out that gene from the virus, because you don't want to try to control an insect with a virus that makes that insect grow a little bigger and eat a little more. So by knocking that out, you can cause those insects to die a little faster, and they don't cause as much damage. So that's one of the
very sneaky
complex things that this virus does in manipulating its host.
Yeah. Hey, one last question. We're getting close to 1 o'clock. Could a deletion of a specific base pair or an addition of one help explain how the event sequence occurs? It's just like a quick mutation, I guess. Not sure.
Yeah, I mean, it would depend on what you're deleting, and I'm not sure what's meant by event sequence means, but typically deleting a single base pair can knock out a gene or change its regulation or that sort of thing. So you can make very small changes and see effects. It's possible, yes.
One last comment from Paul. What we learn from work like this is Gary's work is it can help us find ways to stop insects from transmitting viruses. And that is very important for all these insect transmitted diseases, viral diseases. So thank you, Gary, for joining us today and telling us about this really fascinating research you're doing. This is something I knew practically nothing about. So this was really cool for me. This is a fun one for me to prepare for. I really enjoyed it. And
I enjoyed talking with you and spreading the word about baculoviruses and insect biology and virus interactions with insects. It's a fascinating area.
It really is. It really is super fascinating. And thank you everyone out there in Zoom world for joining us. And please join us for BTI's next Breaking Ground discussion series is on Tuesday, June 21st with Dr. Joyce Van Eck, who uses biotechnological approaches to study gene function and crop improvement. And she was actually our very first guest way back in June 2020. So it'll be nice to have her back after exactly 2 years. Our second year anniversary, we'll have Joyce back. That'll be great. And she's always great, so please don't miss it. And you can go to btiscience.org to register. And I'm going to put a bunch of URLs in the chat box for you all that I will be reading out, so you'll have them right there nice and easy. And you can read more about BTI's current research and many other neat stories about BTI science in our annual report, which you can find online at btiscience.org/annual report. And BTI is an independent nonprofit research institute, and we operate in large part thanks to the generosity of community members like you. So if you'd like to make a gift to support BTI, you can donate online at btiscience.org/give, or email our development team at development@btiscience.org. And we really appreciate all your support. And thank you so much. These are really fun events. I really enjoy doing them, and I'm glad people keep coming to them. It seems like you enjoy them as well. So thank you so much for your interest and support of Boyce Thompson Institute, and have a wonderful day, and please be well. Bye, everyone.